can't hear you, but it's very nice to see you.
Yeah, good morning, Manjula.
Good morning,
In
ma'am.
fact, good evening to you there in US. Hope you're all doing well. So we are very glad and happy to have you here on the international webinar. Thank you so much for accepting our invitation and taking
My
out your
pleasure, ma'am.
valuable time to be with us. I think the students
My
are going to
pleasure.
get immensely, benefited by your talk, and then the light you're going to shed on the research, latest research in the area of COVID. Thank you so much.
Thank you. It's my pleasure too. Thank you so much.
Thank you. Thank you.
Good morning, one and all. I'm Swathi Ramachandran from third year BSc Microbiology Biochemistry Chemistry, Sree Vidyanikethan Degree College, Tirupati. On behalf of Department of Biological Sciences, I welcome you all for the international webinar on recent advances in COVID research. Now, I request our Honorable Principal, Dr. D. Ashalatha ma'am, to give a welcome speech. Over to you, ma'am.
Good morning, everybody. Indeed, it's a great pleasure to once again welcome you all for the international webinar. And this is specifically on the recent advances in the COVID research. See, all of us, we are very actually excited to be part of this new year 2021. With the kind of an adventure that we had in 2020, I think all the students and staff at Sree Vidyanikethan Degree College did progress well because we had a series of excellent intellectual engagements for all of us in the form of webinars and student and faculty interaction programs. So on the behalf of the Department of Biological Sciences and Sree Vidyanikethan Group of Educational Institutions and Sree Vidyanikethan Degree College, I welcome everybody for this international webinar. And I am very much thankful sincerely to the management, our Honorable Chairperson, Padmasree Dr. Mohan Babu Garu, and our revered CEO, Sri Vishnu Garu, for always being there for us and supporting us in all our endeavors. And great thanks, or all the onus of this program goes to the excellent team of faculty and students who have made this day possible. So I think this is a kind of a start that we wanted to have for the new year, and we actually did so many kinds of activities in the previous months that this is going to start up a new avenue of research that we are going to take up in the coming up days. So to start with, we all know that we have Dr. Manjula with us here, joining us all the way from Pennsylvania, US. Ma'am, so much glad and actually honored to have you here as our guest speaker for this international webinar, a valuable resource. And I hope that our students are going to get immensely benefited by your talk and by your research avenues, kind of light that you're going to shed on it. So we all know that COVID has made us very strong. Indeed, we are all greatly indebted to COVID to make ourselves think twice, thrice, what is going to be the sustainable, living of a human being in this world to come. So we all know that even though we have been hearing a lot that the tweaking or the production of the vaccines, or maybe the rolling out of the vaccines, is taking months of time, we know that there has been a considerable research going on in order to make human life very safe. But then, not only tackling the retroviruses, which is not so very easy when it comes to the clinical trials and research. We know that the shedding of the viral, transmission, or the reduction of the viral transmission is much more important nowadays rather than protecting a human being from COVID. We knew that, or we were able to understand that somebody who is healthy with body, mind, and soul, and following very good food habits, are very less on the respiratory side of kind of diseases. They were able to take up the COVID in a positive, different way because they were able to fight it out. Then we were seeing that many of the people who were very unfortunate that they could not sustain and they could not fight it out, and we have lost them as well. But then at this juncture, I would actually like to emphasize that we have excellent people like Dr. Manjula, who have been working in the area of research, youngsters who have been surging forward to take this world and make them say, see, AVM BioMed is the place where Madam works, and I've heard a lot about it because excellent universities and colleges across the world, they work with AVM BioMed because they do a lot of preclinical trials as well as biomarker specialization trials. So I think Madam is going to give us a great insight into the programs that they are dealing with, not only with the COVID research, with the latest advances that we can look at, because it's not only about keeping the human being safe, but also cutting down of a lot of things. But I think this COVID has given a great, kind of a break to the research value, because in the last 100 years, when we see, we had more viruses and more diseases than vaccines and medicines. So I think all the pharmaceutical companies and the biomed companies across the world, they've got a boom kind of a, happiness, because they are ready now in order to tell the world that what exactly research can do. Thank you so much, Madam, for being part of this great, it's not a war, but it's a kind of a battle that we are all doing with to make each and every one of our future generation, all the youngsters safe, all the kids safe, and the entire future of our world safe. Thank you for being so much involved in research, and I hope that with your valuable suggestions, words, and the light that you're going to shed on the research value, which is going to be part of this future of science and technology, not only science but technology, our children will learn and take up sustainable as well as progressive research in the college as well. Thank you everyone for giving me this opportunity, and I hope that you will have a great session. Thank you all once again. God bless all of us.
Thank you, ma'am. Now, I request Mahesh P. from second year BSc Microbiology, Biochemistry, Chemistry, Sree Vidyanikethan Degree College, to introduce today's eminent resource person. Over to you, Mahesh.
Good morning, sir. Good morning, madam. Dr. Manjula Mummadisetti is a senior scientist at AVM BioMed in Pennsylvania, USA, where she heads the proteomic mass spectrometry unit. Here, she works in early cancer biomarker discovery along with the solving proteomic problem for different universities and pharmaceutical industries. Previously, Dr. Mummadisetti worked as postdoctoral researcher at Ruger University in New Jersey. Here, she studied the mechanism of biomineral formation in corals. Her findings on the biomineral proteins will be useful to understand the corals reef better and impact of climatic changes on the corals. Education. Dr. Manjula Mummadisetti earned her PhD from Louisiana State University, Louisiana, USA. Here, she research work has pivotal in understanding the interaction of proteins related to photosystem 2. These proteins are essential for the survival of plants, and hence the understanding of their interactions are important for the field of photosynthesis as well as artificial photosynthesis. This research was also featured in the Science News. Prior to this, she completed her BSc in microbiology, botany and chemistry, and MSc in biochemistry from Osmania University, Hyderabad. Currently, Dr. Manjula Mummadisetti is highly invested in science outreach. She currently serves as assistant editor for journal Bioprotocol and has been a scientific reviewer for more than 10 scientific journals. She also serves as judge every year for the high school's state level science fair called as New Jersey North Regional Science Fair, organized by Nokia Bell Labs. Over to Swathi.
Thank you, Mahesh. I deem it an honor to invite today's esteemed resource person, Dr. Manjula Mummadisetti, to enlighten us about the recent advances in COVID research. All participants are eagerly waiting to listen to you, ma'am.
Thank you so much. First of all, it's my absolute pleasure to be here, ma'am. Ashalatha, ma'am. Thanks a lot for inviting me, coordinating the entire event. And thanks a lot to Ravikanth sir, Lekhya Priya ma'am, Mahesh for reaching out to me, and then constantly coordinating the time because students had exams and stuff, but they really were patient enough to have this event organized finally. So I'm actually delighted to be here. I have always been involved in reaching out to students and help in any way possible. So this is actually great because in this time, it's important to talk about COVID-19. Last year, we all have seen it's so much going on. All of a sudden, there was shutdown, lockdown. We all couldn't move out of the house. So it's important to talk, to understand the science behind it. And I have made the presentation in such a way, since I understand it is for undergraduate students. So it's important for me that all of you understand the science behind it, what is going on, the new vaccines that are out. And at the same time, I would also like to talk to you today about how could you make your undergraduate time here more successful by maybe reading more scientific journals or be able to write something that you could use in future. So my talk would be divided into two parts. My first part would be about COVID research. The second part would be about more on the scientific writing. So I'm happy to be here. Thanks a lot to all the students who are going to be patient for another 1 hour and listen to me. So let's look at some of the research. Let me see if I can share my screen. Okay, is it visible?
Yeah, it is
Yeah.
visible.
Yes, yes, madam.
It's F5 so that we can
Yes,
see.
sir.
Good thing, yeah.
Okay, great. So to Mahesh and to Ashalatha ma'am, it was a very nice introduction about me. I'm also going to leave a contact information if any of your students would like to reach out to me on LinkedIn. You could connect with me. You could ask any questions. You can have any scientific discussion. I'm happy for any of those. So we are going to talk about COVID, which is caused by the virus, which is called SARS-CoV-2. I'm going to give a little bit of terminology here. The virus is SARS-CoV-2, and this is the disease, COVID-19. I want to give a little bit of difference between epidemic versus pandemic. Epidemic is confined to a smaller area or a particular region. Pandemic is an epidemic that occurs worldwide and covers international boundaries. So basically, pandemic is everywhere in the world. You guys are making history. Remember that, because you are part of this pandemic and you are cruising along really well. Okay, so you have seen these numbers, not to scare you, but it's important for all of us to remember how many people have suffered across the world. So it's like in the entire world, as of February 9th, this month, globally, 106 million cases were confirmed. In India, there were 10 million. Deaths in US were, you can see globally, it's 2 million. In India itself, 152,000. Recovered 59 million. In India, 10 lakhs, if I were to speak in lakhs, 10 million, sorry. So many people are affected. It's important for us to see how we could contribute to our safety as well as the safety of the people around us. I want to give a little bit of statistics here in terms of the age group. So anybody with the age group over 65 years, you can see this is rate per 100,000 population and then the time. So anybody with the age group over 65 years, they are the maximum affected. Anybody 50 to 64, you can see they are next. And then 18 to 49, less than these two. And then anybody less than that, less than 17 years is this. Although these are the numbers comparing a large population, but there have been cases where people less than 18-year-old have also suffered and shown like critical cases. Okay, this is another interesting thing comparing different ethnicity. So white people shown in black, in green, black people in, I don't know which color you're seeing, pink or red. And then Asian, I'm going to focus, which includes us, is light green. So percentage of people, their age groups. So if you compare white people, it increases with the age. And then in case of Asian, 0 to 4 years, 5 to 17, 18 to 49, 50 to 64. Pretty much we are doing really well. So there is something really right in our race that we aren't as affected with age when compared to other ethnicity. Okay, I'm going to get into the science behind this virus, a little bit about the structure. This virus is an enveloped virus. It's an RNA virus. We have heard so much about it, but let's dig a little bit deeper. So since you all are undergrads, I'm assuming all of you have heard about lipid bilayer. Fluid mosaic model, I remember studying that in my 11th class. So I'm assuming you would all have heard about it. So this is a lipid bilayer. It's all biological membranes are made up of lipid bilayers, basically. And biological membranes not only have lipids, they can have proteins, they can have glycolipids, cholesterol, all of that stuff. So for COVID or SARS-CoV-2 virus, it has spike proteins, it has membrane proteins embedded in the membrane, M protein, it has nucleocapsid protein, and then small envelope protein.
Okay, very good.
Okay, so spike protein is the reason for all the infection that we are seeing. It mediates receptor binding as well as fusion, which we will see in a minute. M protein and small envelope protein, these are both involved in viral assembly. And then E protein is also important for the release of the virus from the human host. Okay, so there are many types of coronaviruses, but there are very few that can infect humans. SARS-CoV-2 is not the first coronavirus that has infected human. There have been previous coronaviruses that have infected human and have only caused mild respiratory problems. Novel coronaviruses are MERS-CoV and SARS-CoV. SARS-CoV happened in 2003, MERS-CoV, Middle East Respiratory Syndrome coronavirus happened in 2012. Because of so much information from these two viruses, today we were able to combat and perform a lot of research, as well as we today have vaccines, only because we were able to fast track a lot of information or research, only because of we knew something from these viruses. Okay, so a little bit on the origin. I'm sure you all know about this. I'll keep it short. So we have heard so much that it all originated in Wuhan. There was a market which was selling, I mean, eating bats is not new. There are many countries that serve bat as food. But then it's possible in the market at some point, this infected, mutated virus, coronavirus bat was also served. Maybe it infected animal, or it's somehow infected and some recombination happened, and eventually the recombination mutant survived in the human host, adapted to it, and that's the reason for the pandemic. Although we do not know the exact source, but we know somewhere there is a relation between bats and Wuhan, and how exactly the series of events happened, we do not know. There are several classes of coronaviruses, alpha, beta, gamma, and delta. Only alpha and beta coronaviruses can infect humans. SARS-CoV is a new coronavirus that emerged through the recombination of bat SARS-related coronavirus, which I just mentioned. The recombined virus infected civets in humans and adapted to these hosts before causing SARS pandemic. The consumption of infected animal as a source of food is a major cause of animal to human transmission of the virus, and due to the close contact with an infected person. Okay. So I'm going to talk a little bit about the entry of virus in human host, how it replicates, and then immune interactions and therapeutics. Therapeutics is really new because all happened in the last 1 or 2 months. Okay, so we just saw the SARS-CoV-2 structure. It has spiky proteins, and then it's made of RNA as its genetic material. So remember how we all studied in our 11th and 12th grade or during undergrad that there is central dogma, DNA to RNA to protein. Now, this is exciting because this one doesn't have DNA as a genetic material. It has RNA as a genetic material. So let's look a little bit on how it goes into a human host or any host and then replicates itself and survives to come out. So this is a human host cell. SARS-CoV, through its spike protein, interacts with a receptor on the host cell. On its interaction, I'm assuming you would have some enzymology class where you would study about the receptor protein interaction. So once those two interact, there is an invagination that forms. There is this entire science behind on how this fusion happens and it comes out, and then the genetic material is released. Viruses are smart. They don't come handy with all the luggage. They carry exactly 3 or 4 proteins that they need, which is coded, and they use the machinery from the host. So host ribosome, the genetic material from the virus, and then that is replicated. It also carries information for certain proteins that I mentioned before. So the genetic material is replicated. This bunch of proteins like nucleocapsid, spike, membrane, and envelope, all of these are, you remember, I'm thinking you would know about protein targeting. So once all of these proteins are made through ER to GC and Golgi complex, and again, all the viral assembly happens and then release exocytosis. So it's straightforward basic biology or protein targeting. You could go brush up your basics, and I'm thinking that would really help you if you wanted to read any research paper related to SARS-CoV. Okay, so one step back, when the spike protein interacts with the receptor protein, basically these receptors are angiotensin converting enzyme or ACE2 receptors. Now, if you were to understand this is a new science, new COVID, all you know is that SARS-CoV-2 interacts with its spike protein to somewhere on the human host. There are some receptors and possibly angiotensin converting enzyme 2 receptors that spike protein interacts with. How would you find out which part of the body is first infected? You would look at where ACE2 receptors are found, right? So last year, somebody looked at, okay, where is the ACE2 expression? This is not new. ACE2 studies have been done before. So it was a matter of going back and looking at where are these ACE2 receptors seen. And all the colored regions are the ones in the body where you would see ACE2 receptors, like heart, liver, intestine, lungs, kidneys, vascular system. Brain does not have it, but recent studies have shown that even brain can get infected. The infection transmission can also happen in brain. Okay, so I talked about the ACE2 receptor. Then there is another protein which is involved in priming, meaning that once it interacts with ACE2, there is another enzyme or protein which is acting as a mediator or cleaving to make the SARS-CoV spike protein more active so that it interacts with ACE2 and then goes through the process of replication and whatnot. I want to compare, remember I mentioned SARS-CoV is the previous infection from 2003, which means there was a lot of research done on SARS-CoV. And these two are so similar that they use very similar receptors, ACE2, TMPR, SS2, and the antibodies that are generated, the neutralizing efficiency by SARS covalescent sera, meaning the antibodies of the SARS infection can also be used for SARS-CoV-2, but it's really moderate effect on neutralizing or functioning against SARS-CoV-2 infection. Okay, so again, going back to the receptor and antigen chemistry, basically, if you have a target cell, spike would interact with receptor. Then there are a bunch of enzymes. Like I was saying, just this interaction is a huge biochemistry. Once these two interact, there is an enzyme that would help, which is called cathepsin L. So these two are internalized in the membrane, and then in the low pH, especially an enveloped virus would need the opening of the envelope and then release of the genetic material. So there is an enzyme that would cleave and cause the low pH would release the genetic material inside. So there is genome expression, the same story. Then there are a bunch of other enzymes that would act and help in the intracellular activation, and then the same release story from the host cell. Okay, I want to touch base a little bit on the spike protein. This is the complete sequence of spike protein. It has a signal peptide. Signal peptide is something which would target to the right place, and it would have to be cleaved off for the mature protein to be active. RBD is receptor binding domain, meaning it's the region of the spike protein that interacts with the receptor on the human host cell. There are a few other regions that we would not be touching upon. This is, you could say, a crystal structure of just the spike region. I want to talk a little bit about a clinical study on very few patients. This is from last year. So they wanted to understand, okay, there are many people getting infected with SARS-CoV-2, but how do we find that these antibodies that are generated, people who are able to survive the SARS-CoV-2 infection, they are generating antibodies, but where are these antibodies actually binding on the virus? So you have to find out what is causing the infection. You have to find the region of the particle, viral particle, that is initiating the infection. And only then you can use that as a drug target or a vaccine target. So these are 3 different colors. There are actually green. This is a different color. This should be a different color. So 3 different patients, 3 different types of antibodies, but all of these were binding to the spike protein in a similar region, in the RBD region. So that's how people came to know, or researchers came to know, RBD is the region, or receptor binding domain is the region that they should be targeting for drugs or for vaccine, anything to fight against SARS-CoV-2. Okay, when there is an interaction between 2 proteins, it is recognized by how tightly they bind, or their binding affinities. So this is comparing the RBD region is present in both SARS-CoV and SARS-CoV-2. So they wanted to, this study is to compare how these two, SARS-CoV-2 versus SARS-CoV, if you had ACE2 receptor, which is the receptor on the human host, which we just saw a few minutes back, how these two interact. So you can see intensities are pretty similar, but there is a much tighter binding. In case of SARS-CoV-2. Okay, I want to bring some attention towards, if you were a scientist and you would be a scientist in future, how would you identify, now you know the sequence of spike protein, you know which region of the spike protein is causing infection in human host cell, but now you want to predict, because it's RNA genome, it is going to mutate. So how would you predict the future and find out what is the next mutation? How do I create a vaccine that would help you to predict any future mutation and consider it so that you have a drug or a vaccine for it? So it's called mutational scanning. RBD region is not very long. I would assume it's like 50 to 100 amino acids long. You could create one residue mutation at a time and then look at the structure. This can all be done computationally, but this was actually an experiment. So any drug that you want to design, you would actually create computational targets and then identify what can be done with it. So mutational scanning, one residue mutation at a time, and then understand RBD and ACE2 protein receptor, their binding affinities. So I'm going to read through this. RBD is the most variable region in coronavirus, obviously, right? Because that's where all the mutations are happening, and that's the region that interacts with the human host receptor. Mutation screen of residues in RBD region is important. Otherwise, we would not be able to predict. Binding affinities with ACE2 calculated. So like I mentioned, that you would have to calculate with each mutation, what is the binding affinity? The tighter it binds, there is a possibility that that mutation would cause higher infection. Suggests important residues affecting the binding affinity and hence may cause immunogenicity. Okay, so I'm going to go a little bit into what happens once the viral particle has infected. So we have been talking about how virus infects, how virus enters the human cell. Now, how does a human cell respond? That's important. I will not give you every single detail of the immune response, but I will touch base on a little bit of a few things. So the first time there is an infection, a new infection, the first thing that gets activated within the first 4 hours to 4 days is like innate immune response. This is not targeted response, but this is basic response where some of the immune cells get activated. After innate immune response occurs, there is more targeted response specific to the antigen, and that's called adaptive immune response. During this time, you have effector cells that are activated, eventually memory phase, which is B cells, which produce antibodies. They are activated. Okay, so comparing the innate and adaptive immune response, if you have not studied about these cells before, it's just an entry or just for you to understand some of these cells exist in your body and these fight as an immune defense against any antigen. So innate immune system consists of a bunch of different cells like dendritic cell, macrophages, mast cells, natural killer, or granulocyte. Once all of these are activated, eventually adaptive immune response, which is different types of T cells and B cells, which produce antibody and also memory, which is how once you have vaccine, any vaccine that you have taken before, it would produce antibodies in your body. And if you see that antigen in future, because there was a memory or there were antibodies, you would be able to reproduce those antibodies again. But again, memory is the key here, which for SARS-CoV-2, unfortunately, we do not have any idea how long our body has a memory of it. Okay, this is interesting. What we see here is antibody-mediated immunity in case of SARS-CoV-2. Virus-specific IgM and IgG are detectable in serum within 7 to 14 days after the onset of symptoms. And as soon as you have IgM and IgG levels increasing, SARS-CoV-2 RNA starts decreasing by day 21, which is why we are told, okay, there is an incubation time initially, and then you would start seeing symptoms during that time. Viral RNA is inversely correlated with neutralizing antibody titers, like I mentioned, IgM and IgG. Higher titers have been observed in critically ill patients. SARS-CoV-1 humoral response is relatively short-lived, and memory B cells may disappear altogether. And this is for the initial SARS-CoV. We do not know about the memory B cells yet for SARS-CoV-2. Okay, so this is an interesting data, like I was mentioning in 2 slides up, that there are different type of B and T cells produced. If somebody wants to, last year, if people wanted to study what type of immune response is produced, or how are people behaving when there is an infection, and that this study uses flow cytometry. Basically, it's a technique that can be used where you can separate cells, like let's say you're using human cells. So if it has, let's say, a fluorescent dye or something on it, like a green fluorescence protein, or let's say it's colored, okay? You have 2 balls which are colored, one is green, one is red. You can separate those based on color. If there is a larger ball, there is a smaller ball, you can separate them based on size. So that's what a flow cytometer does. It would separate cells based on its properties. So some of these patients had activated B and T cells, meaning that this machine could sort the patient's blood based on B and T cells. They were also able to see a lot of CD4 T cells produced versus very few CD8 T cells. The differences between CD4, 8, and T is one of them is like activating the immune response. The other one is just a helper. The basis of this study is basically being able to see at what stage are you producing which type of cells, immune cells. So I think this study helped a lot in understanding at what stage, how is our body behaving. I have in my slides, I have included a lot of clinical study information just for you to understand in a certain condition, how would you find a research problem? How would you find an answer to a research problem? Okay, so I mentioned about the similarity between SARS-CoV and SARS-CoV-2. Imagine how much time it takes for a vaccine to be produced. It takes really long time. For us, it's only been a year. This SARS-CoV-2 information came out in December 2019. By March, it was considered a pandemic. So all the researchers, anybody that could contribute towards SARS-CoV-2 research, they made themselves available and started working on it. So it's not even March today, yet we are still in Feb. In less than a year, we have so many vaccines from so many different companies. The reason being the similarity between the SARS-CoV infection that was seen first in 2003 versus SARS-CoV-2 infection that we are seeing today. So much information was already available. Genome sequencing, the technology has changed so much. I will show you a sequence. So this is a protein, basically. I'm sorry, just 1 minute. So this is a comparison between SARS-CoV-2, SARS-CoV, and MERS-CoV, where you are looking at the protein sequence of the spike protein. And there is a huge similarity between SARS-CoV-2 and SARS-CoV, these two. MERS-CoV is not really similar. So all the green regions are like a match of individual residues. I'm hoping that you all know proteins are made of amino acids, and you can align the amino acids to know how similar they are. So this is a similarity. You can actually see, I'm not too sure if you have heard about Western blot. It's a way to detect if a protein is present or not. So basically in this Western blot, they were looking at receptor binding domain, interacting region. And in MERS-CoV, SARS-CoV RBD specific antibodies could not find anything in the MERS, and it's found in these two. Basically, just for you to understand that these two are highly similar and antibodies could detect these two easily. Okay, so I'm going to talk a little bit about vaccines. This is my slide from last year when I was giving a talk on COVID-19. This was not very long ago, and there were so many vaccines which were done by different companies. Okay, so I'm going to talk about very few vaccines today, which is viral vaccines can be made either activated, inactivated, or live attenuated. Um, so inactivated is you either like reduce its immunogenicity by either heat or some form inactivation of the viruses, or you could use a chemical like uh, okay, I'm not going to throw big words here, but you can inactivate the viruses. And because its structure is still the same, remember I mentioned receptor and spike protein interaction. So although since the structure is same, even if the virus is dead, it is going to produce infection, or it would raise the immune response because the virus is still considered an antigen or an outside, a foreign body, which our body would detect. So using that information, the only problem with these viral vaccines is that it needs production of large amounts of viruses. Stricter working conditions, biosafety level 3, just know that there are different safety level processes that you have to use, especially if you are using any infectious organisms. Um, Extensive safety testing would be required to ensure that these viruses do not revert to infective forms. However, if you use these type of vaccines, these are more like you can use it in like you provide one shot of the vaccine and it would provide much longer response. Examples of viral vaccines is Covaxin, um, and this is in India, and I'm very sure that you all know about it. So immune cells can still recognize the virus, although it's dead, but because immune cells can recognize it, it would make antibodies. Same with Covishield, it's slightly different. It's made from the weakened common cold virus or adenovirus, and I'm thinking you would eventually learn about it in your viral biology. So adenovirus is interesting. We all have seen adenovirus, so many different forms of it in our life, maybe 70, 80 adenoviruses that have infected us before. So interestingly, if you have to come up with this method of using adenovirus, you would have to come up with a virus that has not infected you before. Such a species would have to be really rare. So Covishield, obviously, it came from chimps. It has been modified to look more like coronavirus, although it cannot cause illness, which is very important. J&J vaccine, Johnson & Johnson, is also similar as weakened common cold virus, adenovirus, but it is coming from human adenovirus. These are replication incompetent. Okay, so one thing is important, and I'm hoping I have a slide for it. Okay, probably not. Okay, so adenovirus is a double-stranded DNA virus. And if you want to provide, because it's a DNA virus and the information of SARS-CoV is in the form of RNA, so you would have to find the DNA sequence, meaning I'm assuming you have studied about genetic code. So DNA to RNA can be translated, and we all know the information, meaning if you have DNA, you can predict the sequence of mRNA. If you can predict the sequence of mRNA, you can predict the sequence of protein. It's same way, if you were to synthesize a protein artificially, you can also predict the sequence of DNA. So since we know the RNA sequence of SARS-CoV-2, you can make DNA sequence out of it, put that sequence in adenovirus, which is a double-stranded DNA. So you can put the viral DNA, which codes for specifically for spike protein, because that's what we need to raise immune response, and put that in adenovirus and have that infect that with human cells. This would express spike protein in the human cells, but there is a catch here. Adenovirus goes in the human cell and then it can replicate. You want to make sure that it infects the human cell, but it should not replicate. It should only infect the cells, release the viral DNA information, and then the viral information should translate into protein and form spike, which would then release, which would raise the immune response and cause antibody production. I'm really hoping I have a slide for it because I really, really think this is important because of Covishield being approved. And India has both Covaxin and Covishield right now. Okay, so weakened virus, inactivated virus, like I mentioned, you have a vaccine, you would have the virus, and then once it infects a cell, the virus would replicate, and then the spiky proteins. So there is something called antigen presenting cell, which would engulf it or which would take it in, and then just the spike proteins are presented on its surface. And because it's a little piece of coronavirus, which is non-self, it would raise an immune response, meaning that our immune system would get activated and find that it's here, and then eventually find ways to activate other immune cells and start the cascade. Okay, so replicating viral vector versus non-replicating viral vector. This is a slide I was talking about. So if you have a replicating viral vector, you have the spike protein, you have viral genes, and this is vaccine pretty much. This is what it is. So it would enter the cell, virus replicates, again, it presents. So it's the same story for adenovirus, but you do not want the adenovirus to replicate because you only want to infect the cell, but you want to, so there are two things going on. If adenovirus is considered as non-self and the immune response is raised against the virus, adenovirus, which is carrying the coronavirus information, you want only the immunogenicity against this particular spike protein, not against the adenovirus. So these vaccines are tricky, but they have been worked out really well. And I'm really happy to be able to share this information with you all. Okay, so DNA vaccine, RNA vaccine, again, the same story, coronavirus spike protein in the rest of the genome, in the rest of the DNA material. You would inject this in the human cell, and then in the nucleus, there is DNA to RNA formation, and DNA to mRNA, and mRNA is again, you would have, it would be translated. And then once it's translated, it would form spike proteins. Then again, this antigen is presented on the surface, and coronavirus spike peptide is again detected for immune response. So basically, any type of vaccine you would design, DNA vaccine or the previous vaccines, they have similar mechanism, but the starting material is different. Okay, so next is the protein-based subunit vaccine, and there are many companies also working on this, although we do not have any approved vaccine for it. So basically, better scalability, easy way of producing large amounts of proteins, because most of these proteins are, when I will give some information on if you were to produce a protein, what you would do. You would just have the piece of DNA, like I mentioned, and put it in bacteria. Bacteria would multiply to any extent, any large amount. You can grow 10 liters of it, 20 liters of it, just collect the protein of your interest. So similarly, and it doesn't have to be bacteria. You can use any other system also. So basically, you can generate any type of protein you want, inject it, and the body would raise the immune response. Okay, so this is different types of vaccines which are approved in addition to the two I mentioned before in India: Pfizer, Moderna, AstraZeneca, Janssen, and Novavax. Interestingly, the number of patients are variable. Pfizer 21,000, 15,000, 4,000 really for AstraZeneca, and then the largest number is for Janssen. Efficacy, one interesting point to note is the number of deaths from COVID-19 after the vaccine was given. during the process of vaccination is none. From vaccination, none. Hospitalizations from COVID-19 during the vaccination, none. Although we say 90% or 100%, 85% efficacy, there was a recent study that showed as long as the efficacy is 50%, that is enough for mild to severe symptoms people, which is interesting because that would be important if we see new mutations in the coronavirus. Okay, so I want to take you through the different steps in vaccine development. Generally, whenever we speak about SARS-CoV related research or any research in university setting, we ignore a lot about what is happening at the clinical trial stage. We talk mostly at the level of what happens on the bench. You would talk about, let's say, how is a microorganism growing, or how is it infecting, or what happens in the body, what techniques to use. Everything on the bench setting is what we generally discuss. What we do not discuss that happens, which takes years and years worth of time, is different stages of vaccine development. So preclinical study generally happens in lab setting. You would use just from culture dishes, growing cells on Petri dishes, to taking it to the animals. And then again, animals is not like one stage. It can go from smaller organisms like mice to rats, to rabbits, to chimpanzees. So there is a really different levels of research that happens before it hits humans. Once you have a vaccine or a drug, what you are really sure through all your lab testing and animal testing, that's when you would give it to humans. And phase 1 clinical trial is really small. It's a small group with like 10 or 15 or 20 people, and you want to test the safety and dosage of the drug or vaccine. Next one would be phase 2 trial, which is a much larger group. Once you have figured that, okay, certain dose is good and it looks safe, you would give it to the next hundreds of people. It could be 100, 200, like few hundreds. Again, you would want to test the safety, immunogenicity, dosage. You might say, well, safety and dosage was tested. It was only tested on 10 people or 20 people. Now you have to use a much larger group because everybody is different. Everybody's human biology is the most complicated. Just because something worked for you may or may not work for another person. So then the next phase is clinical, phase 3 clinical trial, which is on thousands of people. And then it includes, again, safety, immunogenicity, and efficacy. After looking at all of this data, you would go to the concerned authority, and here it's FDA, Food and Drug Administration, and Center for Disease Control. You would go for approval, and then once it is approved, it hits the market, people are taking it. Even then, we do not leave the drugs just because it's approved. That does not mean that everybody can take it. There is a group that would constantly keep looking at what are the problems. Although thousands of people were fine with it, now are we able to use it in different countries? Is everybody in the world able to take it? If anybody has reported any symptoms, new symptoms, or any deaths or anything, then at any stage of after approval, a drug can still be retracted back, or a vaccine can still be retracted back. So phase 4 is still collecting information to ensure continued safety and effectiveness after approval of a vaccine or a drug. Okay. So it's very interesting that for different vaccines that we have seen so far over the years, and I'm not giving any example here, but it would take anywhere from 8 to 10 to 15 years. But here, in this case, for SARS-CoV-2, we have been extremely quick. In 1 year, we have a vaccine. not a vaccine, but many vaccines. Why is that so? There was so much talk that I was hearing, many people being concerned about, can we even trust the vaccines? Why not, is the question. The first thing is, the main reason for everything so be fast tracked is because many of the steps were done simultaneously. So SARS-CoV-2 genome released, it says January 2020, but I think it was in December. I'm not certain. Preclinical studies started immediately. Phase 1-2 trials were done simultaneously. 2 and 3 for many companies were done simultaneously, and some countries approved vaccine for emergency use, but with much caution. And then all of these, as soon as we had some data, generally for vaccines, you would want to release that data to everyone. So if you have phase 2 trial information or phase 3 trial information, generally you would wait until it's published to the public. But I think most of the vaccines for SARS-CoV-2, we do not wait so much. As long as there is data, it's slowly released and published, but immediately companies started their next trial process as soon as they had all the data and statistically analyzed and all of that. I think that helped a lot. A lot of times there are different regulatory boards which are involved. There is money and funding issues, but this time really all the organizations possible, governments, they pushed in, they made sure that any time delays that can happen is avoided. Only science and trials are done. Okay, so I'm getting close to what I wanted to speak about COVID. Really what we all have to make sure is ensure the safety of yourself and ensure the safety of your surrounding people. Stay at home, self-isolate, avoid touching, you know the story. Always wear mask, clean your hands frequently, and then maintain physical distance of at least a meter. So one interesting thing that I wanted to bring up is if you are really close to, like if you are in a closed environment and there are multiple people, many people sitting together, what we have been doing at work, what I have been doing at work is use 2 masks at a time. And that way you're certain that even if there is air conditioner, we are not infecting anybody else or we are not getting infected by anybody else. Cover your nose and mouth. That's very important whenever you cough and sneeze. Stay away from crowds as much as possible. I know many people have been saying now the SARS-CoV-2 infection is dying down, but it's very important that we do not care just for ourselves, our age groups, but it's also important that you care for your parents' generation, your grandparents' generation. Regularly clean and disinfect. Do not forget these steps. Make sure that you're constantly following. I know many of us in younger generation, we tend to avoid many of these, which I've seen at least here in the people around me, but make sure these are really important, not just for yourself, but also for the people around you. Okay, so that's about the COVID research. I took you guys through a little bit of basics on the COVID SARS-CoV-2 structure, how it can infect, and the vaccines that are new and out and available for us to use. I think what I want to understand from the group that is listening to me now is, do you want to ask questions now, or do you want me to go through the rest of the presentation and talk to you about scientific research and scientific writing?
Yeah, you can go ahead, no problem.
Okay,
Students
thank you,
are
sir.
preparing the questions, and once you wind it up, they will start asking the questions. They have already
Okay.
noted down some questions.
Okay, sure. Thank you. So I wanted a little bit of interactive session with maybe one of the coordinator or anybody. What is everyone's BSc group here?
It is microbiology, biochemistry, and chemistry.
Okay, microbiology, biochemistry, and chemistry. That's wonderful. So I think what I wanted to tell everyone here is I have also been an undergrad before. And whenever I was a BSc student, I realized that I was not very certain what to do next. Um, I wasn't sure if I wanted to study master's. I wasn't sure if I wanted to study PhD. And I'm very sure not every single person is the same, meaning that some of you might still be in dilemma. What, where do we want to study next? Do we want to study in India, abroad, this college, which college? Do we really want to go up to PhD? What are the job prospects, and so on and so forth. But in all of this, um, between all of these questions, I think what you have to remember is, what is your passion? Are you really scientifically inclined? Do you want to study science in the future? If you want to study science, it's also possible that in your high school you really liked math, you really liked physics, you liked chemistry or over biology. Maybe you like microbiology more over biochemistry. What do you want to study next? I think the answer lies in you identifying what you like based on reading, based on you doing the research. The more you read, it's not somebody else suggesting you what you want to do next. It's for you yourself to find out what you want to do next. Um, Even if you were to study, let's say you were very good at physics and you think, okay, microbiology is too much of microorganisms for me at this point. What I want you all to know is biology is an umbrella term. Principles of physics are used in biophysics. Chemistry is very important to understand biochemistry. Math is used in generating mathematical problems to study organism behavior patterns, population changes over time, any statistics you want to do. Or, okay, another example, we talked about clinical trials. If you want to study clinical trials is done by biologists, right? You would imagine so. But you also have to generate all the data that's obtained. You want to make sense out of it. You can be a biostatistician understanding all of that data. You can generate mathematical models to understand all the patterns in it. Let's say you want to know there was a clinical trial going on and that data did not consider age group or certain aspects. For example, there was a cancer patient that's included in the clinical trial, and there is some other infection or disease person related information. All the clinical trials are not the same, but you can, this is called metadata. You can get the information from the existing clinical trial or existing data in research community or published somewhere, extracting that data out and making sense out of it. You can use mathematical models there. You being a biologist, you can do that. Statistics, like I mentioned, using large data set, information technology, coding, you can use that for computational biology and bioinformatics, or be part of science policy making process. Like literally anything that you have heard of, you can apply that to biology. I remember when I was a PhD student, I was using a lot of mass spectrometry. This is used a lot in the industry by chemists. This was what was told to me by a friend of mine. And I was mentioning, well, I'm handling that in my lab and I'm a biologist. So the understanding of the terminology is not important. How you apply it towards a biological question is what is important. So I think what is important for all of you is to figure out what interests you the most and then take a track in that direction. But if you have not understood which track you want to take, you will never be able to even get in. If you want to study MSc and you ask another person, what do I study? It is not going to help you. You have to figure out what is your passion, what is your obsession, what is really making you move forward. It can be biology, it can be any of these fields, or even not. Okay, so it is very interesting. I was having a conversation with Ravikanth sir, and I was very much impressed that he was talking about this topic of undergraduate writing and research. If I had somebody told me this is important, I would have made an effort, because if I want to study even master's, let's say, not even PhD, somebody looking at your application and saying, oh, this person has published during their undergrad, your application will be 10 times stronger than anybody else. Even if you scored 70% in your undergrad and somebody else scoring 90%, but you have a research paper published, you will be weighing, your application will be weighing a lot more stronger, because outright, there are not many people, not many undergraduates that publish. So not only as a, you would be making a mark in scientific field, at the same time, you would be learning a lot more while reading scientific papers. You would learn science, you would learn more English in general. Reading capability would increase, writing capability would increase, scientific writing capability would increase. So I think what I want all of you to understand is, during this journey in your undergrad program, being able to learn more science, figuring out what science you enjoy, being able to write any of the scientific paper. Just pick up anything and write a review article, read 2 or 3 scientific papers and write a summary of it. Start from there. It would really, really help you. And maybe form groups of 4 or 5. Come up with a bunch of ideas and start writing. If you guys write a piece that is worth publishing, it can be cleaned up. The piece can be cleaned up by any of your teachers. And then I'm happy to help in any way I can. During this research and writing process, you would make new connections, because let's say you're reading something, some scientific paper you didn't understand, you can actually reach out to that particular author. You can reach out to somebody in colleges or universities and say, well, I read about a bunch of these things, and this is really attractive, whatever you're working on. You can make new connections that way with different professors. And you never know, you might find an internship there. You might look for fellowships either in India or outside of India and perform some undergraduate research with different people. That is about undergraduate research. First and foremost thing is, there is summer coming up. Maybe look up for summer internships, work with any professor, any lab, little experiment that you are able to contribute towards. That would be amazing. But I don't think anybody would let you enter unless you have the basics of their research. So the first step is reading before you even ask for some internship. Writing. So like I mentioned, it really makes your application stronger for higher education. When I was applying for my PhD, if I had a paper, during my interview, it would really have added a few extra points. I want to add one more thing. I generally, when Mahesh was giving a little bit of introduction about me, a lot of high school students start their research right from high school here. They spend their full year evenings working with different professors, different PhD students, and then they have a nice poster, which we end up judging, which is equivalent to something like a master's student work. And it's amazing that they have the aptitude to read, to understand, to design experiments. And I think it would be wonderful if you are able to get there. And that would only happen if you start somewhere from reading and writing and reaching out to people. It obviously helps to learn and develop scientific aptitude. So let's say you start writing or reading. Where would you even start, right? I mean, it's not easy to start writing a research paper and publishing. Now, you really have to know a lot more than just where you are today. So I think I have jotted down a bunch of places that you could consider. Excuse me. So you could publish in either any of the local journals. You could If you have a college newsletter, or if you're able to start a newsletter, write content for that. Blog, I mean, we all are social media people now, and anybody can create their own blog. Maybe create a blog for your own class and then start writing different topics once per month or whatever you can do, you know. One interesting thing that I've seen in many people here is if you want to learn the writing skills, you can go on Quora. We all read Quora, right? You can just go there. There are many people that post scientific questions, non-scientific questions, anything. Start writing there. Try to use new words or any new word that you see while reading a paper or newspaper, whatever. Try to use that. And that will also help you with your writing skills. This is something that you should look up: medical writing, regulatory writing. It's a writing skill as well as writing in terms of something like vaccines or clinical trials or whatever, something on those lines. If you are interested more in drug-related studies, so write something content related to that. Any promotional literature. If there was a drug, if there was a vaccine, let's say Covaxin, if you were to write a promotional literature, how would you write about that? Look up a bunch of things, how they are written, and then write something related to that. There are many companies that would be willing to hire you if you're good at this. Writing content for newspapers. This is something that I had done as a high schooler in India. I would every week send my little piece of writing to Times of India. Sometimes it would be picked, sometimes it would not be picked. Sometimes it wouldn't go in my age group, then I would send it to a younger age group through my brother, which is not good, but at least if it gets accepted, you get that little motivation. Bottom line is, unless you write, unless you read, you wouldn't know, but writing is an important skill. Any field you go to, let's say you decide today you want to study masters, or let's say you want to go for civils exams or banking, or you decide to get into IT. You never know what step you want to take next. Either or, if you have good writing skills, it's a means of communication. You need to have a good, cleaned, refined skill of writing, which goes really a long way during all of our careers. Okay, so I want to talk a little bit about how to write or how to read a scientific paper. I'm very certain at some point your teachers would tell you this, but if you were to start something sooner before that session happens, I would say start with reading scientific review articles. Let's say you got interested looking at SARS-CoV-2, my talk today. You can go back and go to googlescholar.com and look up all the review articles that are possible. You might be overwhelmed with the amount of data that's available. Then sort it based on any specific, let's say, cholesterol and SARS-CoV-2 or cancer and SARS-CoV-2. All the review articles that show up, you can put that together. Okay, start with one paper at a time. Once you have a paper, read the abstract first. If it's a research article, you may not always understand the abstract, which is when you should go to introduction. Read the introduction. The way introduction is written is it would be really broad for everyone's understanding, and slowly they will start bringing it closer to what they have as the main content. So if you are able to understand introduction, that's an amazing step. You can say kudos to yourself if you're able to understand introduction, because that's the first step to understand the paper. Then directly jump to discussion. If you're able to understand discussion, that's amazing. Do not read the materials and methods or results, anything, because you're not at that stage yet. But once you have read introduction and discussion, there will be some certain things about the results saying that we use this technique, we use these proteins or this DNA or that technique, whatever. Then you can go back to results and start trying to understand. Anything that you do not understand, you can obviously go to your teachers or anybody and they will help you. So that's the first step. Read review article. If you're reading any of these scientific pieces, start with introduction and discussion. If you decide to write something, go in the same order. Write introduction first. Once you have done that, figure out what would be your discussion, and then you can fill the rest of it. I want to share some examples of what you could think about writing being an undergrad today. I think what is important for you, which I really see now, I'm not sure how many of you have decided to go applying for masters. I know when I was applying for masters, I started preparing for the entrance exams at least a year and a half before or something like that. If you are also there already, maybe this is the time that you start considering having at least one paper published. This will really, really, really help you. You may not have decided about PhD, but once you hit masters, if you enjoyed it, I know that you would be trying to get into the PhD world. You would want to get your PhD. But regardless of whether you get master's or PhD or not, you do not want to lose this time that you already have today. So try as much as possible. Let's say if you were writing a review article, we can discuss more about this another day. The first step, like I mentioned, is read a bunch of review articles to understand it. They are much simpler. But if you're writing a research article, since you're not in a research lab, since you do not work in a research lab, you're not in a research setting yet, the way you can think about is consider training yourself in analyzing data, meaning, let's say something is available. There are 2 or 3 different papers. Looking at that, are you able to come up with some hypothesis that can be studied just by sitting, just by looking at, for example, can you statistically analyze something? Can you do bioinformatic analysis of certain things? If you think you can perform some bioinformatic analysis or some statistical analysis, you can do that, sitting at your home, and then write a story related to that. Once you are there, you can reach out to me or to any of your teachers, and we'll help you to write that piece together, scientific piece. So I think what I really want to stress on is spend some time, figure out what you enjoy reading, start from there. Spend time every week on reading some review articles. Make a few groups in the class and come up with a bunch of different ideas, and then combine and decide on a topic and write something related to that. If you are able to write something, I'm very certain you can get it published somewhere. It can be something like a zero impact. All these journals are graded based on zero impact to like really nature and science. We don't have to target nature and science today. We can target something like close to between 0 and 1 impact, let's say. But if you have something published, it will really be impactful in your career. I cannot say more. I really wish somebody had told me this when I was studying my undergrad. With that, I think we have come to the end of COVID as well as writing topic. If you have any questions, please go for it. I do have some questions for all the students, but I'll take my turn after you all are done. With that, I want to thank Principal Ma'am. Thanks a lot for organizing. Dr. Ravikanth sir, Lekhya ma'am, and Mahesh, who reached out to me initially once he heard my talk, my previous COVID talk. Thank you.
Thank you so much, ma'am, for sharing your valuable knowledge. Now, I request Pragna Chinmayee from 3rd year Microbiology, Biochemistry and Chemistry to start the question and answer session. Over to you, Pragna.
Good morning, ma'am. Good morning, ma'am.
Good morning. Very good morning.
I have few questions for you. Okay, sir. Question number 1. There was a news that SARS-CoV-2 is made in the laboratory in China. How far is it true?
It's an interesting question that comes again and again. I do not believe that it would have come from the lab. There was a paper that also came how evolutionarily or how it would have transitioned from bats and then adapted to human hosts. But I do not believe that it has come from lab. We do not know the exact origin, but it's not something that we all should believe or think about.
What's the value and validity of coronavirus antibody testing?
I'm sorry, can you repeat that?
What is the value and validity of coronavirus antibody testing?
What is the value of antibody testing? What do you mean by that question? I'm sorry, can you please help me understand?
Validity of coronavirus antibody testing.
What is the validity
Yes, ma'am.
of coronavirus antibody testing? So you want to understand?
Yeah, to what extent was it successful? Because it has been tried on some patients, tried on some people. So that is what she means to ask.
Okay,
What was the population on which it was tested?
Madam, to add to it, there was a lot of plasma testing also going on, and they were actually thinking about whether it could neutralize the retroviruses. So I think they are asking the question in that direction.
Okay. I'm sorry, since the voice was breaking when the first person spoke. So this is when you mentioned, so antibody testing, you have to remember how the SARS-CoV is working, okay? So many of the people are able to generate antibodies. Some people are not generating antibodies. There is a constant timeline that you have to remember. Once antibodies are generated, you have to go for testing in a certain period of time. So there is a lot of variable here. One is the antibody getting detected? Have you gone and gotten tested in the right time? And the third thing is, have you even produced the antibodies, or is there an escape mechanism going on inside? So has it been successful? Yes, for the majority of people, it has been successful. But for some of them, it's not visible. So is it 100% successful? No, none of these tests are 100% successful. So to answer yes or no is not something that I can say, but it has been successful for a majority of people. But for many people, it is not because of different variables that are here. Does that answer your question?
Yes, ma'am. How advanced and useful is RNA interference in production of vaccines and treat other diseases?
Okay, you're still breaking.
How advanced and useful is RNA interference in production of vaccine and treat other diseases?
RNA interference. Okay, that's an interesting method in the treatment. RNAi treatments can be used, but in this case, we really have to see what is available today. RNAi method is interesting because you can, break. But then I think what is important to know is what is available, what is functional in terms of the vaccines that are available now. A lot of times, it's not the method that is important, but is it scalable? That is more important. Does that answer the question?
Yes, ma'am. How can furin inhibitors can be targeted as potential drug therapies for SARS-CoV?
Furin inhibitors. So furin inhibitors are basically one of the enzymes that is involved in priming. Is that correct?
Yes, ma'am.
Okay. So if you were to use furin inhibitors, I think many companies are considering that. It is a great way of assuming how effective it is something we would have to see over time. But it comes in the pathway of the virus infecting the host cell. So if you can stop at any step for the viral entry, it is great. But again, are we able to find the right drug? Are we able to scale it up? That's something that we would have to consider.
Based on what kind of clinical factors the COVID vaccine dosage was prescribed for different age groups?
I'm sorry. So based on what factors dosage was considered?
Yeah.
Based on what factors dosage was considered? So basically, I have not been involved with the clinical trials. I can look up the answer, but generally, whenever you want to see the dosage, there is a different, for example, in a lab setting, if you were to provide a dosage to, let's say, rats, you would want to see the life span that has changed, infectivity that is affected, and then how much. So for example, you have cells, how many cells, human cells are dying versus how much immune response is raised and how much of the virus is dying. So there are 2 or 3 things that you would have to consider in any time you would be testing in a lab, So however, for humans, it's different. I've not been involved with the clinical trials myself. So I might have to look that up and answer it later.
Adenovirus vaccine is used as coronavirus vaccine. Are the serotype 4 as the first shot and serotype 7 as second shot? Is it?
I could only hear adenovirus.
Adenoviruses now vaccine is used as coronavirus vaccine. Is the serotype 4 as the first shot and the serotype 7 as the second shot? Because the serotype 4 and 7 are already stopped manufacturing in 1999.
Okay, again, these questions are something that I do not have expertise in. I do know that some of these are old, but are these good or not? This is out of my expertise, but I can look it up and answer it for you.
Thank you. Do we need to improvise the vaccine every time because the modification is observed from place to place and time to time?
That's an interesting question that we have all been battling with. So again, to understand, you mentioned that if each time there is a mutation, do we have to come up with a new vaccine? Is that the question you asked?
Yes, ma'am.
Okay, sorry. Since you have been breaking, I'm just trying to understand. I'm repeating it. Okay, so this is a problem, right? It's the same thing with influenza virus. Each time you see a mutation, which is why there are so many flu vaccines coming out every year. In US, we take flu vaccines every year, but are they effective or not? That is a separate question. It's the same thing with the current vaccine. If there is a mutation in SARS-CoV-2, what would we be doing? Would we be taking new vaccines? The answer is we do not know yet. The mutation would have to be in the region that causes infection. It has to be in the spike protein somewhere. RBD region, there are other regions. Are they really causing the infection that is needed? So it has to happen at the structural level. It has to happen on the spike protein. Assuming that it does happen on the spike protein, are the antibodies, are the vaccines that are available today, are they able to neutralize them? That's another thing. Most of the vaccines that have come out, for example, the 2 new mutations that we know about, one is from the South Africa and one is from UK. There are a lot of studies that are coming out that most of these vaccines are able to reduce the effect of these mutations, the vaccines that are available, like from Pfizer or from Johnson & Johnson. But how much is still a question. So there is a lot of unknown related to it. We have to understand that we cannot panic that there is a mutation coming. What we have to understand, it has to be at the structural level. There is one example that I can provide, which came out in the recent Nature paper, probably a week back, which was a cancer patient who already had lower immune and lower immunity, basically, and was already under heavy drugs and whatnot, had COVID infection or SARS-CoV-2 infection. And this person was given convalescent sera from somebody who has recovered. What happened is in first 10 days, this person recovered from SARS-CoV-2 infection. However, within those 10 days, there was another mutation in the virus that happened, which caused infection in a different way, because now the coronavirus has infection which happened at spike protein. The antibodies are unable to neutralize it, and his immune system is down, is unable to fight with this new virus. This person died immediately. So initially, his immune response was going like linearly high, and then suddenly it drops because the new virus, he is unable to fight with them. So he died in like less than 2 weeks time. So again, the question is what mutation it is. Are these vaccines able to fight with it? Most of the vaccines are derived or made in a way that it considers multiple mutations in the spike protein region, or the region that has to be considered. But will it be able to take care of all possible mutations? We don't have an answer to that today. I'm sorry, I went a little longer, but that's the answer. We don't have an answer to it.
Okay, ma'am. What is the size, the populations on which the vaccine was tested so far? Have anybody had any side effects reported?
There are side effects. Yes, there are side effects. We should all look at the published material, which is very important. However, the way clinical studies are performed, like I was talking about the statistics involved, you always look at the effect, the positive effect of neutralizing antibodies in killing SARS-CoV-2, let's say, versus the side effects that are there. So the way clinical trials are designed is not based on one side effects that's there. You always have to gauge between side effects versus the plus parts of it, the benefits of it. So you have to consider these side effects change with mild versus critical. Okay, so you have to see what side effects are there. Should you be taking it or not? Is it mild? Is it critical? What is the genetic background, or is this person having anything else, like any other disease going on? So can we go back to your question again? You mentioned about what are the side effects? Is that question?
Is the size, the populations on which the vaccine was tested so far?
What is the population size for which the vaccine is tested so far?
Yes, ma'am.
Okay, so one of my slide mentioned about that. The population size is several thousands for different clinical trials for different companies. Anywhere from 4,000 to 5,000 to 20,000 to 25,000 people for phase 3.
As SARS-CoV, the alpha and the beta MERS-CoV has already appeared in the year 2017 and caused about death over 600. Why not people were conscious? I mean, the scientist or the former fellow person, even the WHO listed it for urgent research and development.
Okay, WHO research, I'm sorry.
The WHO listed it for urgent research and development as the beta virus MERS-CoV has already appeared in the year 2017 and caused over 600 deaths.
Okay, what's the question? I'm sorry.
This virus appeared way back in 2017. Why scientists and other pharma people were not interested, or why did they take up?
Okay. 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 So what appeared before in 2003, SARS-CoV? What appeared in 2012 or 17 for MERS is completely different from what is happening today. Those are much on the epidemic scale. People reacting to it was much different. People reacting to SARS-CoV-2 is much different. The number of deaths, the most reaction, although all of these coronaviruses that have infected humans come in the category of upper respiratory tract infection, but they were much on the milder side. It was not like many people are getting affected with it. So it was really not something of serious matter, although there were many deaths with it, but it was not at this scale. So obviously, if somebody went with that infection, they were treated. But today's case is completely different. You can see that it has crossed the borders and many countries are affected. So obviously, if government had to take any decision at that point, it would be much different at that point. It would be much less scalable.
Thank you so much, ma'am. Now, I request our head, Department of Biological Sciences, Dr. S.V. Ravikanth, sir, to propose vote of thanks. Over to you, sir.
Dear Dr. Manjula, at the outset, on behalf of the organizing committee and all of us, I have to apologize to you for the inadvertent delay. It
Not
took
at all.
almost more than a couple of months for us to finalize this program. And I'm extremely sorry for all this delay, which was not in
Not
our
at all.
hands. I hope you will understand that. I would like to thank our beloved principal for all her encouragement right from the beginning. She has been encouraging us, and we could conduct this webinar successfully. I would like to thank my colleagues, Dr. Lekhya, Priya and Dr. Sree Devi, for their help in organizing this webinar. I also thank Mrs. Srivani, who helped me in designing this Google Forms and all that. I would also like to thank our media team for all their help. I'm really, really grateful to you because you chose two topics, and very happy with the word umbrella term referred to biology. I think we can also add English to that because scientific writing and publishing research articles requires good communication skills. I'm thankful to Mr. Mahesh for introducing Dr. Manjula to all of us. It was thoroughly professional. You also mentioned about internship, fellowships, and the newsletters. I'm pretty sure students will do that. And really particularly impressed with the thank you slide. I have not seen such a slide for a long, long time. It included the name of the principal, myself, and my colleagues and students. That was a brilliant idea. And I am sure people like you will contribute a lot to the scientific field in course of time. Next time when you visit India, we request you to come to Sree Vidyanikethan Degree College and have a visit. Suggest some good measures and hope we will have some MOUs with you and we will take this forward. Now I request our beloved Principal Madam to give her remarks.
Thank you, sir. In fact, Manjula ma'am, it was a pleasure listening to your lecture and all your thought process, which was, as sir said, thoroughly professional. I think this is the day the students should actually learn like how they have to be very much, professional in the way that they prepare for the future, as well as presentations, or how they look at learning in the best possible way. Thank you so much for joining us. In fact, I enjoyed each and every explanation that you were giving for every slide, because the moment you started, that itself, it shows that you have done, a great work when you are doing your learning, as well as now contributing towards research. And one more thing, what I've observed is that, and not only from your profile, but even from this presentation, is that you have love towards learning, and you often mention the word called reading, which I think the students have to adopt from today to actually take it up, very seriously. And one more thing is that I've seen from your resume also that you are very much connected to the student community, and you have been judging a lot of research programs or the research fairs or the science fairs that you have been associated with in US as well. So that's a great gesture. See, being part of the professional career and then contributing and actually sharing it with people, it's a great gesture. I think people like you, we need them more in this world to make the students motivated to take up learning as a serious, kind of an, activity, as I would say, not only an activity, a kind of an, investment into the future. So I hope that the children would have learned how a best student, a best researcher can contribute to the world's, not only in the pandemic situations or the learning processes, and also different kinds of developments that we are going to see to make ourselves a very safe kind of a planet, including the environmental conservation that we are going to do, keeping up all these issues which we are tackling right now. Thank you so much, madam. Once again, we wish you a very wonderful year ahead and a prosperous future. And I hope, as sir said, you will be in touch with us and we'll collaborate to do many more exciting events. Thank you so much. God bless you.
Thanks a lot. It was a pleasure to be here. Anything that I could help with in any of these aspects, anything that I could not answer, or if you have any other questions, please message it to Dr. Ravikanth sir, and I'll be happy to respond to those. I do have a few questions for students. If you want me to send it to Ravikanth sir, I would do so, or I can ask now if there are a few extra minutes.
Please, ma'am, please, you can ask them.
Okay. Okay. Well, I wasn't going to go. Okay, that's the slide for students. How many of you know which one was the first vaccine?
Penicillin was the first vaccine.
Yeah, for some reason, I can't hear.
Ma'am, to my knowledge, penicillin was the first vaccine. Is that correct?
Do you know how was the smallpox discovered? Okay. Okay, never mind. Please do look up about the Louis Pasteur story. I think it would be very interesting for you to know everything that I talked about so far. You would be able to relate to the vaccine development process. The other thing I wanted to ask, we talked so much extensively about the receptor and antigen interaction and how your body would respond to it. I think what I want you to know or learn about is how would one identify such an interaction from a research perspective. The reason I'm having this questionnaire session is not for you all to feel competitive about it. You don't have to answer it today. You don't have to answer it right now. But go back and search for it, Google it on if there is a new virus, there is a new interaction that you have to study. How would you study it? What experiments would you do to study such an interaction? Look it up. How would you study protein interaction? How would you study a drug versus like a host cell receptor interaction? Look up that aspect. The reason I say that is because I talked a lot about scientific writing, and such things will be really helpful for you to gauge a paper, whether if the experiments were done right or not, or can you do something like that. I also want to come to question number 3, which is, why is RNA replication not mutation proof? You know how DNA replication is mutation proof? Why is RNA replication not mutation proof? Why are we even talking about mutations, right? So look it up. You don't have to answer me right now. I want to take you through the bioinformatics process a little bit. And I'm hoping that these slides would be available to you somewhere, or I can send it to Ravikanth sir, and you can actually go step by step and try different things. So what I want you to do is, now that we have talked so extensively about vaccines, my homework to you is to go look up the structure of the antibody. Okay, it has constant regions, it has variable regions, or you can look up any protein. It doesn't have to be antibody. But answer for yourself, what regions do these antibodies contain? Okay, how are those different? Can you compare one antibody to another antibody? How are they different? This is something basic bioinformatics that you can do sitting at your home. All you need is a computer with an internet. You don't need anybody's help. It's really basic. Google it and you'll find. This is an important exercise if you were to analyze two separate papers and you want to try something new and put it as a scientific piece, you would be able to do that. This would be your first step. Let's say you have the antibody, you know which regions are present. Go to UniProt. It is a site where you can look up all proteins, okay? Find its FASTA sequence, which is basically the protein sequence in a certain format. Find it, download it. And then now that you have one sequence, maybe just an UniProt type IgM, IgA, IgE, anything, or IgM from human, from something else, you'll have, as soon as you type IgM and UniProt, you'll have like thousands of responses. Click on all of those. Let's say take top 5, okay? Download those and then go find how to align the sequences. FASTA is the protein sequence. Find out how to align them. I'm not sure if you have done this exercise or not. If you have not, this is an important exercise for you to know how to compare different proteins. A little bit of biochemistry because you have biochemistry as a subject. Then once you have done the multiple sequence alignment, I want you to go to PDB, which is Protein Database Bank. Okay, any protein that you study, if you want to know its 3D structure, you know, all of us, everything that exists has a 3D structure if you can resolve it, right? Proteins are really small, they have 3D structures. 2D is like X and Y, 3D is Z direction. Proteins structures in their 3 dimension can be resolved by many different methods. Okay, X-ray crystallography, cryo-EM, anything. Okay, so go to PDB, find any protein of your interest, or let's say IgG. Okay, find the structure of it, download that PDB file, and then go to PyMol, which is another program. So see, I'm teaching you multiple programs. Go through those. This will be an interesting exercise. And this is a structure viewer, a 3D structure viewer. You can open the protein or IgG or anything that you want to see and play with it. You know, it's a 3D structure. You can play with the cursor. It will move in different directions. And then try to learn this particular program. Next, you know, to enjoy this process, you can then go to XPESI. Now you have studied different steps of protein expression, protein translation, the post-translational modifications. You can go to xpesi.com and look at this particular protein of interest and find which different post-translational modifications occur on that particular protein. And I think just with this journey, you would have learned a lot. Anytime you learn about a new protein in biochemistry, go do this exercise and you will learn so much more. Whenever you look up in UniProt or anywhere, any specific protein sequence, it also tells you what different regions it has, what different interacting partners or any specific binding domains. So I think do that for at least one protein and then start reading anything related to that, and that will be very helpful. So that's a few questions and homework from me. Okay, with that, I can say thanks a lot for the opportunity and for the wonderful words from Principal Ma'am and from Ravikanth sir.
Thanks a lot, madam. And I'm pretty sure they will take up this as an assignment and they will submit it to me in about a week's time. It will help them to write some research articles, or at least it will lay some foundation as to how you have to go ahead when you are writing a research article. Thanks a lot, madam.
Absolutely. Thank you.
Any questions? Thank you, madam. Have a nice day.
Thank you. You too have a wonderful day, sir.
Thank you, madam. Thank you so much. Thank you.
Bye.
I will end the session.
Yes,
now.
sir. Yeah, please, please.
Good night, Manju. Thanks a lot.